my work here. Okay, so temperature is one of the most fundamental processes that shapes life on Earth, and especially for ectothermic organisms whose body temperature closely tracks the surrounding environment. So the environmental temperature determines their metabolic rate, which influences their life history traits, and consequently where their populations can persist and how they perform. So temperature has a strong impact across all levels of biological organization, from the molecular interactions of individual proteins to organismal traits like birth and death and feeding rates, up to population level metrics like abundance, and then all the way up to macroecological patterns like range limits. And in fact, temperature is one of the key factors that links patterns across all of these biological scales. And with average global temperatures increasing due to climate change, there's been a lot of concern over how warmer temperatures are going to affect transmission of infectious diseases. So this is the climate barcode made by Ed Hawkins and the Climate Lab book. And it's a really striking visualization showing that annual global temperatures have increased by almost 1.5 degrees over the past 180 years. But answering this question for how temperature affects transmission of infectious diseases is actually difficult to answer for a few reasons. So first, disease transmission is a really complex process. So the response of transmission to temperature depends on the thermal biology of the host, the parasite or pathogen, and how they interact together. So we need to be able to account for all of these different processes or traits that depend on temperature and contribute to disease transmission. And additionally, there's often tension between different traits that are impacted by temperature in different ways. So we need a way to quantify and kind of weigh these different effects together. And second, the thermal responses of biological traits are usually nonlinear and often unimodal. So where they peak at an optimal temperature somewhere in the middle, and then decrease at colder and hotter temperatures to these minimum and maximum thresholds. And so one important consequence of this specific type of nonlinearity is that an increase in temperature will have a different effect depending on what your current temperature is. So an increase in temperature could lead to an increase in the trait value, or the same increase in temperature could lead to a decrease in the trait value. So we need to be really careful when we're characterizing these curves and making predictions for temperatures that we don't have direct observations of, as is often the case for climate change. So today I'm going to talk about some of my work on mosquito-borne pathogens, which have become one of the best model systems for understanding how temperature affects transmission of infectious disease. And our general approach has been to use trait-based ecology, where we fit thermal response curves to data from lab experiments for all of the important mosquito and pathogen traits. Then we use a mathematical model to calculate how R0, which is the disease reproduction number, should respond to temperature based on those trait thermal responses. And so all of you are probably more familiar with R0 than you were before the COVID pandemic, but it's basically the average number of secondary infections that you expect from a single primary infection, assuming that everyone is susceptible to infection. And so R0 should be proportional to disease transmission in many cases. Then we have validated those R0 models with field data of real infections. And then we can use those R0 models to generate maps that will allow us to visualize the temperature, how temperature shapes transmission risk, and make predictions for the impacts of climate change. So today I'm just going to talk about these first two steps, the trait thermal responses and the R0 model. And our aim in the past has been to apply the same approach to as many different mosquito-borne pathogen systems as possible in order to be able to make comparisons across different vector species and pathogens in an apples to apples kind of way. And so we've now done this for 16 different vector pathogen systems, 11 which I was leading, and they were synthesized last year in Ecology letters. So now that we've done that, I've been revisiting some of the decisions that we made while we were developing this approach. And so before I get into my new work, I kind of need to summarize a little bit of the old work so we can understand what those assumptions were. And so before I get into that, I just want to acknowledge some of the key people who I've worked with on this project and who worked to generate some of the models that I'm going to present. I particularly want to highlight Aaron Mordecai, who's my former postdoc advisor at Stanford, and who led this whole mega project. And of course, Van Savage, who's my current advisor, and who's been working with me on the new results that I'm going to present. So in order for mosquito-borne diseases to be transmitted, mosquitoes have to be present and abundant. They need to bite an infected host. They then need to survive long enough that the pathogen can complete its development inside the mosquito and migrate to the salivary glands. And then the mosquito needs to bite an uninfected host to pass that pathogen on. And so the mathematical model for R0 can be divided conceptually into three different components of this transmission cycle. And each of these components can be expressed as a function of mosquito and pathogen traits that depend on temperature. So mosquito density depends on fecundity, the development rate of mosquitoes, and the survival at all life stages. The contact rate between mosquitoes and the host depends on how often they're biting, the biting rate. And then the probability that a mosquito becomes infectious depends on how quickly the pathogen develops inside the mosquito relative to how long adult mosquitoes live, as well as the vector competence, which is the maximum proportion of mosquitoes that will become infectious, assuming that they all live long enough. So kind of taking the time component out. And so for each of these traits, we fit nonlinear thermal responses describing how they respond to temperature. And we use two different functions, the Briere that's asymmetrical and left skewed, and a rearranged version of the quadratic that is symmetrical. And both of these functions have three parameters, this constant Q, and then the thermal limits, the Tmin and Tmax. So then we plug these trait thermal responses into the Ross-McDonald model for R0 for mosquito-borne diseases. And you can see our three main components in the numerator here. So we have mosquito density, we have the contact rate or biting rate of mosquitoes, and it's squared since they have to bite twice, once to acquire the infection and once to pass it on. And then the probability of becoming infectious highlighted in that box there. And then there's an additional term for adult mortality in the denominator that accounts for infectious mosquitoes dying. And so we've also extended the standard Ross-McDonald R0 model to substitute an expression for mosquito density as a function of demographic parameters for the mosquitoes. So this expression was first developed by Mordecai et al. 2013, and it was based on work by Parham and Michael 2010. And because this model is isolating just the effect of temperature on mosquito and pathogen physiology, when we get the results, we scale them between 0 and 1 to get a relative R0 where everything is compared to the maximum transmission allowed by temperature. So the absolute value of R naught depends on other factors besides temperature that we aren't considering here. And a bonus is that it makes it easier to compare between the different vector and pathogen systems. So here are the results for relative R naught that show these thermal responses across the different vector and pathogen systems. And this approach emphasizes the importance of three key values: the optimum, where transmission is maximized, and then the thermal limits at the cold and hot ends, where temperatures should prevent transmission from occurring. And so here's another way of viewing those same results that show the 95% credible intervals for each of those key temperature values: the optimum here in the middle, the lower thermal limit, and the upper thermal limit. And then here's an example of the way that we can use those R naught models to generate maps of transmission risk and predictions for how climate change will affect transmission. So here I'm showing the population at risk for malaria under climate change, and you can see that the hot spot for transmission we think is going to shift from West Africa. As it gets too hot in West Africa, it's going to move to the East African highlands. So that's the old stuff. And so what I'm really here to talk about today is there a better way to calculate mosquito density? What is actually driving that probability of becoming infectious? And how good are metabolic theory of ecology-based approaches at approximating R naught? And so before I dive in, I should probably tell you what metabolic theory of ecology is. So it's a theory that uses basic principles from physics to generate a set of very general and simple predictions for how biological processes scale with temperature, as well as mass, but we're going to be ignoring the mass side of things here. And so this is the classic paper for metabolic theory of ecology, Brown et al., 2004. I just want to point out that Van was also on this paper. So the key attributes of metabolic theory that are relevant for my applications are that it focuses on processes that scale with basal metabolic rate. It tries to build an intuitive understanding for how different processes scale together. And it uses a specific set of thermal response functions. And so the examples that I'm going to talk about today are just 3 of the 16 models that we put together. So it's dengue virus in Aedes aegypti, here at the bottom, malaria in Anopheles vectors, and then West Nile virus in Culex pipiens. And I selected these pairs because they're some of the most important diseases, and they also show quite a bit of variety in their thermal responses. So for question number 1, is there a better way to calculate mosquito density? We're going to turn to a metabolic theory-based approach for estimating population growth rate, or little r. So here's the old equation that I've already showed you for mosquito density. It's the one that Mordecai et al. Developed based on the work from Parham and Michael. And it contains the traits fecundity, the percentage of larvae or eggs that survive to adults, and the mosquito development rate, and the daily adult mortality rate. And it's based on basically just intuition about the birth rate of new adults into the population. And this new method, which comes from a paper by UCLA faculty, Priyanka Amarsakari and Van Savage, is derived from Euler's equation, and it has most of the same traits with one exception. Because metabolic theory makes predictions about rates rather than proportions, instead of having the percent of larvae that make it to adulthood, it has the daily juvenile mortality rate, kind of as a match to the daily adult mortality rate. And then there's also this W here, which isn't a parameter, it's a mathematical function called Lambert's W. Here I'm showing the results for population growth rate instead of mosquito density, just because I think it's a little bit more intuitive for most biologists to look at the numerical values. And the results show that this new method for predicting population growth rate or mosquito density gives results that are more asymmetrical and much more reasonable based on what we think real numerical values for population growth rate should be. There are still important differences between the different species, but the scale of these differences is reduced compared to the old method. And so here I'm just kind of zooming in so you can really see that on the old method panel there. And I think the answer here is clearly yes. This is a much better way to calculate the predicted mosquito population growth rate and mosquito density. And so in terms of how this change impacts our predicted R naught, there are no changes in the thermal limits, but the optimal temperature is shifted to almost a degree warmer. And there's a big decrease in the absolute magnitude of predicted R naught, which gives us actually much more realistic values. So that was actually another one of the reasons why we were scaling our results between 0 and 1, was just because we didn't actually think that the maximum R naught here should be 20, or in some cases, over 100. So I was pretty happy when I saw these results. And I'm just showing the results here for dengue, but they're qualitatively similar for all three species, or all three vector pathogen pairs. Okay, so now I want to look at what drives the probability of becoming infectious. I'm showing that mathematical expression for R naught here again. And so let's break down this probability of becoming infectious a bit. And I want to focus just by looking at this part, which is the probability of an adult mosquito surviving long enough for the pathogen to complete development. So here's the function for e to the negative x, where x is the ratio of adult mortality rate and pathogen development rate. Or it might be easier to think of it as a ratio of times the average adult lifespan and the time it takes for the pathogen to develop. And so when the average lifespan of a mosquito is equal to the pathogen development time here at x equals 1, about 37% of mosquitoes survive this development period, and so could potentially become infectious. And then, of course, as that ratio changes, if development time is shorter relative to adult lifespan, we have more becoming infectious. If development time is longer compared to adult lifespan, we have fewer becoming infectious. And if we look at our three example systems, the relationship between adult mortality rate here in solid and the pathogen development rate in dashed, and how much these two thermal responses overlap, we can see that it varies quite a bit between these different example systems, which translates into substantial differences in the thermal responses of their ratio and the proportion of mosquitoes, or the proportion of mosquitoes that are expected to survive the pathogen development time. And then when we add in this vector competence term, interestingly, it acts as kind of an equalizer, and we end up with curves that are much more similar to each other than they were over here. And I also think it's pretty interesting from a metabolic theory perspective that this final proportion that become infectious can be right skewed, left skewed, or quite symmetrical, depending on what the response of vector competence is, and how that interacts with this proportion that survived the infectious period. Okay, so now moving on to the final question, how good are metabolic theory-based approaches at approximating R0? So I'm gonna have to explain this problem a little bit. It's basically that metabolic theory is often proposed as a way to model data poor systems, but basically all of the published case studies that use metabolic theory to make this argument are really data rich. And this makes sense because the metabolic theory thermal response functions have 4 to 6 parameters, and they can be difficult to fit, especially without good data. And additionally, many of these thermal response functions have to be fit in Kelvin rather than Celsius, which biologists are not super used to or comfortable with. But on the other hand, many of the parameters in these thermal response functions have characteristic values, like the activation energies, or easy biological interpretations for things like the optimum temperature and thermal breadth. And so this means that in theory, we should be able to make informed guesses based on minimal data. It's just that no one has done this in the published literature yet. And then I also want to point out that one of the major differences between these metabolic theory-based thermal response curves and the previous approaches that we've been using to model R0 for mosquito-borne diseases is that they approach zero much more gradually. And so I'll show you what I mean by that. Here are the 3 parameter functions that we've been using in the past. The Briere for the left skewed traits and the quadratic for the more symmetrical traits. And both of these functions really emphasize the thermal limits, the Tmin and the Tmax. And in fact, those are 2 of the 3 parameters in these equations. But these limits occur where the organisms are pretty unhappy, and biological traits are actually the hardest to measure, and biologists don't often like doing experiments at these really extreme temperatures just because the organisms are dying all the time. In contrast, the metabolic theory-based functions approach zero much more gradually since they're based on the basal metabolic rate, and your metabolic rate is never really zero unless you're literally dead. But you can see this like sharp Schoolfield function is pretty nasty. So what I was interested in seeing is how well we could approximate R0 using these metabolic theory-based thermal responses estimated just by capturing the general shape of the data where these traits are the easiest to measure. So basically, if we just know where the trait optimum is and we have some idea for how it increases on both sides, how bad is our estimate of R0 going to be if we don't really have good estimates for like where these thermal limits are here? And the answer is actually much closer than I anticipated. So it's a pretty close match. And although the thermal limits are off by 2 to 3 degrees, our efforts to validate these R0 models for mosquito-borne disease with field data show much stronger evidence for the general shape and for the optimum temperature than they do for the thermal limits. And we think that this is likely due to the fact that there's a lot of temperature variation in nature that occurs over various timescales compared to the constant temperature in the lab settings where these trait data come from. So based on this result, I'm actually much more optimistic about the prospect of using metabolic theory for data poor systems than I was before I started this work. I was actually like very much a skeptic for certain aspects of metabolic theory. So conclusions, is there a better way to estimate mosquito density than the methods we've been using? Yes, definitely. By using that analytical expression for population growth rate, we can get more realistic estimates. It doesn't affect the thermal limits, and it yields slightly warmer optima. What drives the probability of becoming infectious for mosquitoes? Vector competence really strongly constrains this value, and it minimizes the big differences in the proportion of mosquitoes that survive the pathogen development time. And then finally, how good are the metabolic theory-based thermal responses that really gradually approach zero at approximating R naught compared to our more data-driven approaches? And they're actually surprisingly good. So with that, I'd like to thank everyone for your attention, and I can take some questions. I realize I went a little bit over, so I apologize for that.